Hello all, hope you are all having a good day. Today we're going to be talking about a molecule called Coprine. This is Coprine. You can see it's got two main parts, chemical moieties. It's got this glutamine right here, and then it's got a cyclopropyl group on the end here. Cyclopropyl groups are fairly rare in nature, so it's always somewhat interesting to find one. This one has an effect on humans. It comes from this Coprinopsis atramentarия, aka the ink cap or Tippler's Bane. It is an edible mushroom with a mild, slightly metallic taste. The dark pigment is used to make ink, and it has the interesting property of being conditionally toxic. And if you're up on your Victorian English, you might have figured out what that condition is. It is toxic only if you drink alcohol with it. Tippler being an old word for drunkard. So, in order to understand how Coprine does this, we need to understand how alcohol metabolism works in the first place. So, let's talk about that. Alcohol metabolism happens in two main biochemical phases. The first one, alcohol dehydrogenase, oxidizes ethanol into acetaldehyde. So, let's go over how it does this. So, the first step is ETOH is this ethanol binding to zinc atom that's being chelated in the enzyme by histidines, I believe. The first step, so the first chemical step of metabolism is an unknown base, think lysine, arginine, histidine, attacks this hydrogen of histidine, so lysine or arginine, attacks this base on hydrogen, removes it from the nitrogen. So that bond breaks, and the electrons start to form a double bond here. Carbon-nitrogen double bond. Now, this carbon, nitrogen double bond right here is going to go ahead and break, put that electron density onto the nitrogen, and nitrogen is then going to attack the hydrogen of serine 48. That is going to lead to that electron density being pushed onto the oxygen. The oxygen is going to push it right into this hydrogen, forming basically reforming that alcohol bond, and leaving this nitrogen or this oxygen to be negatively charged, being stabilized by zinc. So the end of all of that leads you to this molecule right here. This situation right here. You've got histidine 51, and then serine 48, and then our ethanol. Alcohol dehydrogenase is then going to bind a cofactor called NAD plus in this case. NAD plus and NADH are used in several biochemical steps, biochemical pathways. They're main components of metabolism, so and energy metabolism. So NAD plus comes up a lot. And what NAD plus is going to do here is it's going to pull away a hydride from this carbon and allow this oxygen, negatively charged oxygen, to form a double bond with the carbon. And after that happens, we get acetaldehyde. This is what acetaldehyde is. And you get the NADH now leaving. So, and then shortly after acetaldehyde leaves, water comes back in and recharges the enzyme. Next step, after you get acetaldehyde, is aldehyde dehydrogenase is going to bind that acetaldehyde, and it's going to bind a cofactor called NADPH, who is basically the same as NADH. It is a hydride source in hydride zinc, and it is used in a lot of biochemical steps. So one of the cysteine residues in aldehyde in aldehyde dehydrogenase is going to attack this acetaldehyde. It's going to form a tetrahedral intermediate with the enzyme, and you're going to get NADPH taking away another hydrogen, or sorry, NADP plus taking away another hydrogen, to form NADPH. At which point, that oxygen is going to push down its lone pair and form a carbon-oxygen double bond. Again, except now you've got a carbon-oxygen double bond, and one of the other bonds is to cysteine, or the sulfur of cysteine, rather. At which point, a glutamate is going to take a water. Water is sort of omnipresent in all of these reactions, and it is going to pull off a hydrogen, and that's going to turn that water into a hydroxide anion. And then that hydroxide anion is going to swiftly attack the carbonyl, and it is going to kick off the cysteine, sulfur, and leave as acetic acid. Then the cysteine is back to its initial state, and water is going to come along and deprotonate the glutamate and return it to its initial state. So, how does Coprine interfere with this? So, Coprine is going to interfere at the second step of alcohol metabolism. What it's going to do is it's going to enter the body, and then in the liver, it's going to get attacked by glutaminase, which is going to form glutamate and 1-amino cyclopropanol. That just means it's got a 3-carbon membered ring, 3 carbons, cyclopropane. It's got an alcohol, and it's got an amino group. So it's 1-amino cyclopropyl alcohol, or cyclopropanol. And then this is going to react with acids and bases in the body, and it's going to form not amino anymore. It's going to form 1-cyclopropanone. It's going to rapidly interconvert between 1-amino cyclopropanone and this hemiacetal, cyclopropyl hemiacetal. But when it is in this cyclopropanone form, it is going to get picked up by aldehyde dehydrogenase, the cysteine, and it is going to and the cysteine is going to attack the cyclopropanone, forming an alcohol right here, and it's going to bind it to the enzyme. Except there's a problem here. You'll note that there are no hydrogens around this carbon center. It's already got 4 bonds, one to oxygen, 2 to different carbons, and then 1 to the enzyme. No hydrogens. So when NADP binds to ALDH, it is not going to see any hydrogen to remove. So the enzyme is going to stay stuck in this spot with this cyclopropyl moiety bound to it irreversibly. And this is going to cause acetaldehyde concentrations in the liver to start spiking. Acetaldehyde is more toxic than ethanol. It is responsible for most of the symptoms of a hangover. So, with alcohol coming in and being oxidized into acetaldehyde by alcohol dehydrogenase, and then not being able to get converted into acetic acid with aldehyde dehydrogenase, because it's all being bound up with coprine, this causes aldehyde concentrations to spike and you to get a hangover really quickly within a few minutes, like 5 to 15 minutes of drinking. And depending on the amount of Coprine and the amount of alcohol you consumed, that could be a hangover that lasts anywhere from hours to days. Coprine lasts about 48 hours in the body. So if you eat Coprine and then within 48 hours drink alcohol, you could poison yourself. Now, I want to take a brief detour away from humans and talk about how Coprinopsis makes this compound. So, frustratingly, this isn't well understood. I wasn't able to find any papers on it. If you are able to find any papers on it, please let me know. But what I'm going to do here is I'm going to show you how a similar compound, 1-amino cyclopropane carboxylic acid, right here, is made. It's part of the Yang cycle, which produces ethylene. Ethylene is very important because it causes plants to ripen. So it's very well so the Yang cycle is really very well understood comparatively. Because if you can control the rates of these enzymes, you can control what speed plants ripen, which means you can keep them shelf-stable for longer, has tons of economic benefits, right? So this 1-amino cyclopropane carboxylic acid, or ACC, is formed from this compound called S-adenosylmethionine, which is a very common molecule in the body. It is mostly used for methyl transfers. This methyl group right here can move around, and to help with the reaction, you're going to get pyridoxal 6-phosphate, also known as vitamin B6. And the first step of the reaction, which isn't being shown here, is this amino group is going to form an imine with pyridoxal 6-phosphate. And it's going to form this carbon-nitrogen double bond while still attached to the S-adenosylmethionine. So, the first step that is shown is a basic residue comes in and pulls off the hydrogen of this carbon right here, which causes a whole chain of arrow pushing, right? It's going to cause this carbon to form a double bond with the nitrogen. It's going to cause this this carbon-nitrogen double bond to break, and this double bond, the electron density is going to go into this carbon-carbon double bond. This carbon-carbon double bond is going to break, and it's going to break resonance here, as it's going to shift into this carbon-carbon double bond, and this which causes this carbon-carbon double bond to break and form a lone pair, become a lone pair on nitrogen. This nitrogen right here. The end of all of that is this situation right here. At which point, it's just going to reverse. So, what happens is this lone pair attacks this carbon, forms a carbon-carbon double bond. This double bond breaks, goes right here, which causes this bond to break and go right here. And it's going to cause this bond to break, become a negative charge on the carbon. Now, you're probably going to say that seems really unlikely. Like this whole setup seems really unlikely. But you have to understand the enzyme is making sure that everything is in the right chemical environment. Everything's being stabilized enough. Everything's being pushed in just the right way to make this reaction happen, right? Then, the next step is where the magic happens. So, this carboanion is going to attack the first thing it sees, and the enzyme is going to make sure the first thing it sees is carbon 4 of S-adenosylmethionine. So it is going to attack carbon 4, and this carbon 4 is then going to boot off this sulfur, and you're going to get the methyl adenosine sulfur portion of the molecule leaving, and you end up with a cyclopropyl group with a carboxylic acid. Then Water is going to come in and it's going to attack this part of this carbon. It's going to kick off the amino group, which is going to cause it to kick off the nitrogen. And then you get 1-amino cyclopropane carboxylic acid leaving the enzyme. So that is just one of many potential routes that Coprinopsis instrumentaria could use to form this 1-amino cyclopropanol. It could use a route very similar to this. As for why it makes coprine, that is also frustratingly well not understood. If you do know why, if you are able to find a paper discussing why Coprinopsis instrumentaria makes coprine in situ, please let me know. It could be used for a variety of things. Those cyclopropyl groups, like I said, ACC degrades into ethylene. So it could be ethylene is made by plants, and it's also made by some fungi. So maybe Tippler's Bane makes it for ethylene. Maybe it's also been found to be insecticidal and antimicrobial. So maybe it's made as a defensive hormone or as a defensive compound for Tippler's Bane. We just don't know. Now, back to humans. You might have had the thought, if coprine locks somebody into a hangover state for hours to days, could we use it to help somebody? To help somebody who has alcohol dependency? Well, it's been investigated, and unfortunately, coprine is a mutagenic in high doses. The doses you would need to give somebody to get them to stay off of alcohol for a long period of time. However, there is a drug called disulfiram that has been used to just this effect. You give disulfiram to somebody, and if they start drinking alcohol again, they poison themselves. It's not lethal, it's not dangerous, it's just very, very uncomfortable. And it can help to break the cycle of abuse in people with alcohol addiction. In any case, that about covers it. That about covers how coprine works and why you should be very careful comparing mushroom soup with wine. Please let me know if there's anything I missed. Thanks for watching and have a stellar day.